Hello, my name is Christy McDonald, and I'm going to discuss two cases for Medical Biochemistry 1, Unit 2 case connections. Let's look at case number 2. Regulation of glucose uptake by the cell. The above brain MRI shows a large glioblastoma tumor with areas of necrosis within the tumor. The tumor is the bright white area within the brain field on the MRI. On the right is a PET scan from the same individual following administration of fluorescently labeled D-glucose. Use the following series of questions to determine how and if glucose uptake differs in the tumor when compared to surrounding tissues. In normal tissue, how is glucose uptake regulated in the following tissues? The liver, skeletal muscle, brain, and adipose tissue. How does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the above image, and why would this be advantageous to the invasive tumor? What modifications may have occurred in the cancer cells to alter glucose uptake as predicted above? First, we were asked how glucose uptake is regulated in the liver, skeletal muscle, brain, and adipose tissue. Starting with the fed state, we see that the liver uses glucose, which is taken up by glucose transporter 2. The uptake of glucose in the liver is insulin independent. The skeletal muscle uses glucose, which is taken up by glucose transporter 4. The uptake of glucose in the skeletal muscles is insulin sensitive. The brain uses glucose, which is taken up by glucose transporters 1 and 3. The uptake of glucose in the brain is insulin independent. And finally, in adipose tissue, glucose is taken up by glucose transporter 4, and the uptake of glucose in adipose tissue is insulin sensitive. In the fasted state, free fatty acids provided from lipolysis are used in the liver and the skeletal muscle. Free fatty acids provided from lipolysis are used as fuels in the liver. In the brain, glucose is provided through gluconeogenesis and glycogenolysis. In adipose tissue, free fatty acids are provided from stored triglycerides. Next, we're asked how glucose uptake in normal tissue compares to glucose uptake by tumor cells in the above image. In both cancer and normal cells, glucose enters glycolysis and through a chain reaction is converted to pyruvate. In normal cells, pyruvate enters the citric acid cycle and through oxidative phosphorylation produces around 38 ATP molecules. However, in cancer cells, we see the Warburg effect. In cancer cells, the cells rely on glycolysis instead of oxidative phosphorylation for energy, even when oxygen is present. Pyruvate is converted to lactate, and production of ATP through oxidative phosphorylation is significantly decreased. Because less ATP is created from each glucose molecule, more glucose is needed by the cancer cells. So why would this be advantageous to the invasive tumor? At first, it seems like a disadvantage for rapidly growing cells. A significantly smaller amount of ATP is being produced. However, by not oxidizing pyruvate through the citric acid cycle, the cancer cells can use some of the molecule for biomass. This means that the cells must use a lot of glucose to get the necessary ATP, but this provides a lot of material to build up biomass for these rapidly growing cells. Finally, we're asked what modifications may have occurred in cancer cells to alter glucose uptake as predicted above. One of the modifications found in cancer cells is the presence of self-sustaining growth factors which cause rapid cell growth. Another modification includes genetic changes in metabolic enzyme encoding genes, metabolite transporters, and other genes. An example is found in the glycolytic pyruvate kinase muscle gene, the PKM. This gene encodes for pyruvate kinase muscle enzyme. PKM1 is found in normal cells, while PKM2 is often found in cancer cells. PKM2 is often found in the low-activity dimeric form. This change allows cancer cells to synthesize macromolecules that are needed for rapid growth. Case 4. Previously healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports she has not given the baby anything to eat other than breast milk since birth. The baby has only recently been sleeping for more than 2 hours between feedings, and that's when the mother reports these symptoms started. Laboratory results are consistent with hypoglycemia, low blood sugar glucose, but are otherwise unremarkable. Epinephrine is administered and there is no increase in blood glucose, but there is an increase in blood lactate. Physical exam reveals a liver edge 4 centimeters below the right intercostal margin. Percussion to the right chest and abdomen confirms hepatomegaly, enlarged liver. Blood glucose levels increase after breastfeeding, but she cannot maintain blood glucose levels within a normal range during fasting. Based on the information above, which of the following is the most likely diagnosis for this patient? A. Hereditary fructose intolerance. B. Galactosemia. C. Glycogen storage disease, inability to use glycogen. Or D. Glucose 6-phosphate dehydrogenase deficiency. I used the U.S. National Library of Medicine to research these diseases. After my research, I chose C, glycogen storage disease, inability to use glycogen, as my answer. Let's look at the options that were dismissed. Option A is hereditary fructose intolerance. Hereditary fructose intolerance is a condition that affects a person's ability to digest fructose, a simple sugar that's mainly found in fruits. Patients with hereditary fructose intolerance develop symptoms when fruits, juices, or other foods containing fructose are introduced into their diet. After ingesting fructose, nausea, bloating, diarrhea, vomiting, and hypoglycemia are commonly experienced. These infants fail to appropriately grow and gain weight, which often is extreme enough to lead to failure to thrive. Repeated ingestion of fructose-containing foods can lead to liver and kidney damage. The liver damage can result in jaundice, an enlarged liver, hepatomegaly, and cirrhosis of the liver. Continued exposure may also result in seizures, coma, and death from liver and kidney failure. The reason that this diagnosis is discredited is because the mother reports she has not given the baby anything to eat other than breast milk since birth. With hereditary fructose intolerance, symptoms occur after ingestion of fructose-containing foods, and fructose is not a natural component of breast milk. Also, no nausea, bloating, abdominal pain, diarrhea, vomiting, or jaundice is noted. Next, we're going to look at galactosemia. Galactosemia affects how galactose is processed. A small amount of galactose is present in many foods. It is primarily part of a larger sugar called lactose, which is found in all dairy products and many baby formulas. The signs and symptoms of galactosemia result from the ability to use galactose to produce energy. In type 1 galactosemia, the most common and most severe form of galactosemia, infants develop life-threatening complications within a few days after birth. These infants typically develop lethargy, jaundice, liver damage, and abnormal bleeding. Galactosemia type 2 and type 3 cause fewer and less severe symptoms, which doesn't explain the patient's presenting symptoms. The reason galactosemia is discredited as a diagnosis is because the symptoms occur shortly after birth with galactosemia babies due to the fact that galactose is in breast milk. Also, no jaundice or abnormal bleeding are noted within this patient. Next, we'll look at answer choice D, glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase deficiency presents almost exclusively in males. This condition causes red blood cells to break down prematurely, which is called hemolysis. The most common symptom associated with glucose 6-phosphate dehydrogenase deficiency is hemolytic anemia, because the red blood cells are destroyed faster than the body can replace them. Hemolytic anemia causes fatigue, shortness of breath, and rapid heart rate. Glucose 6-phosphate dehydrogenase deficiency also causes mild to severe jaundice in newborns. Many people with this disorder, however, never experience any symptoms, and they are unaware they even have the condition. The reason that this diagnosis is discredited is because it occurs almost exclusively in males, and there was no hemolytic anemia or jaundice noted in this baby. Now we'll go back and look at the chosen answer, C, glycogen storage disease, the inability to use glycogen, which is likely the von Guericke disease. Von Guericke disease is an inherited disorder caused by the buildup of glycogen in the body's cells. The buildup of glycogen in the liver, kidneys, and small intestines inhibits the organ's ability to function normally. Symptoms typically appear around 3 or 4 months old. Around this time, all infants start sleeping through the night and do not eat as frequently. Infants with von Guericke's disease may develop hypoglycemia, which can lead to seizures. Patients can also develop lactic acidosis, high blood levels of uric acid or hyperuricemia, and excess amounts of fats in the blood, hyperlipidemia. Patients also develop an enlarged liver and kidneys. They experience diarrhea and deposits of cholesterol in the skin. One of the tests that can be used to diagnose von Guericke disease is the epinephrine challenge test. When epinephrine is administered, the glucose levels show little to no increase, however, lactate levels do increase. The glucose storage disease is indicated because the disease has not shown symptoms until the baby was 2 months old, just after starting to sleep through the night. Seizures explains the jittery spells, the fasting hypoglycemia, increased lactate with epinephrine, and hepatomegaly. The references are on the slides that follow.